So guys, when I was talking about the electron transport chain and the proton gradient in ATP formation during the process of photosynthesis, or specifically during our discussion on the process of photophosphorylation, did something struck you? Did something struck your mind? Yeah? Have you heard these terms? Protein complexes, electron carriers, electron donors, protein gradient, movement of electron, synthesis of ATP, chemiosmosis. I mean, you must have heard. Yes, we have talked a lot about this when we were talking about, yes, electron transport chain which takes place in the mitochondria. So let's compare between the electron transport chain which is taking place in the mitochondria and which take place in the plants in the chloroplast. First we see the similarities. First of all, the membrane, it is a membrane associated transport. I mean, the chain, the complexes, they are embedded into the membranes of these organelles. In the case of, for instance, mitochondria is the inner mitochondrial membrane, and in the case of chloroplast, it's the thylakoid membranes. Of course, there is a creation of proton gradient. The creation of proton gradient in both cases is formed because of the movement of the electrons. And not only the proton gradient is the same, I mean the formation of proton gradient is similar, but also one other similarity is that the fate, actual use of the proton gradient is also the similar. In both cases, you want to make ATP through the action of ATP synthase. But there are differences also which we have to keep in mind. First of all is the source of electrons. For photosynthesis, the source of electron is the water. Of course, first of all, the electrons are activated by the excitation of the light. But the electron which even come into the photosystem too, they are because of the splitting of the water. So the source of electron in photosynthesis is water. In oxidative phosphorylation, which takes place in the mitochondria, it is the NADH, FADH2, which you get from Krebs cycle or glycolytic pathway. Direction of proton pumping is also interesting here. For photosynthesis, it takes place, the pumping of the protons, which you want to eventually make a proton gradient. In photosynthesis, the pumping takes place, or pumping leads to the movement of protons into the thylakoid membrane or thylakoid membrane space in the chloroplast. While in oxidative phosphorylation, it takes the protons outside the matrix of the mitochondria. Here it takes outside the matrix of the mitochondria. Here it takes inside the thylakoid membrane in photosynthesis. So once you have pumped the protons, then you want to move them down the hill to get the energy for making ATP. So in photosynthesis, the protons are then moved down the hill out of the thylakoid space, while in oxidative phosphorylation, it goes into the matrix. So the protons move inside to the matrix in the case of oxidative phosphorylation, while for photosynthesis, it goes out of the thylakoid membrane. But the purpose is the same, of course, that you get a gradient of proton, which will eventually be used in the formation of ATP. Yes, terminal acceptor of the electron in the case of photosynthesis is NADP, which eventually gives you NADPH, which will be used in the Calvin cycle. While in oxidative phosphorylation, the terminal carrier of the electron is the oxygen, which will be converted into water. So again, for water, as I have already told you, is the electron source. The final destination of electron is the NADP and NADPH2. I have already told you. While in the case of photosynthesis, in case of mitochondria, at the end of the day, it is the water which is formed because of the reception of electrons by oxygen. So these are the similarities and these are the differences. I mean, you can go through it. I mean, it's not difficult. But what I really want you to think a little bit, that what is it? I mean, on one extreme is a chloroplast, a very different organelle. I mean, it's in plants only and not in animals. And on the other hand, in animals, we have mitochondria, which is also in the plants. But still, such similar sort of structures are involved in the making of what we see, the energy. So why there is so much similarities? Doesn't it struck you? The oxidative phosphorylation and the photophosphorylation, does it have an origin? Can we say they are linked with each other in very ancient times? Maybe during the very start of life? Maybe you should think about it. And I have already told you that in biological sciences, you have to think the things in the context of evolution, that how chemical processes are linked with each other, how physiological processes are linked with each other, how the species are linked with each other. Because then you will really get the concept in the best way. Thank you.